All right, so moving on to the hallmarks of cancer. Okay, so what did we say about the hallmarks now? So the what's if we talk about how cancers, when we say cancer, we're saying it's a malignant neoplasm, malignant tumor. Okay, so how do they come about? How do cancers arise? What makes a single cell go bad ab initio? What starts one cell, a normal cell, on that road to become a cancer? Okay, you have your normal cell here. What's the thing that starts it off and makes it become sort of, or let me use the term evil, an evil cell that now starts disobeying, doing whatever it wants to do, causing havoc for the body? DNA mutations, right? Like I said, most cancers are caused by mutations in the DNA. So we said DNA, when we get to biochemistry next week, DNA is that structure. If you have your cell, okay, two main parts. It has the cell cytoplasm. Okay, the cell has composed of cytoplasm and then the nucleus. Okay, nucleus is usually the blue or purple part in the cell. So, and inside the nucleus, what is there? Your DNA, right? The DNA is like the master house of the coordinator of the cell. Just the same way, if you the best way I like to describe it is, if you think of your DNA, it's like the White House where the president is. You're going to the US, so it's like the White House where the president is, and every other thing else is the most important structure in the US there, because that's where all the activities of the government are coordinated. All right, it's the one that directs everything that happens in that country. So the same thing, the DNA is the one that directs everything that happens in the cell. And so once that DNA gets corrupted, that means that cell it becomes an abnormal cell. All right, becomes an abnormal cell, and if it doesn't die off, if the mutation doesn't kill the cell, it becomes an abnormal cell that can start doing abnormal things, which will transform it into a cancer. Okay. All right. So once a cell becomes a cancer, what are those features? What are those features of a cancer uh that that that's that explain what are the features of a cancer that explain how it's able to grow uncontrollably, eventually okay, invade and metastasize. Okay, so growing uncontrollably and then it invades, metastasize. That's what we're talking about, this hallmark of cancers now. All right. These are the hallmark of cancer that we're talking about. So all of this describes all the features that a cancer cell or the cancers have that allows them to grow uncontrollably, invade, behave abnormally, and eventually spread to distant organs. All right. So starting with the first one, so there are about about 8 of them. So that's the first one: growth, survival, growth signal self-sufficiency. Second, anti-growth signal insensitivity. The third is evasion of apoptosis. Fourth is limitless replicative potential. Fifth is sustained angiogenesis. Sixth is Warburg effect. Seventh, invasion, immune evasion in cancers. Eight, tumor invasion. Nine, metastasis. Okay, so nine, not eight, sorry. All right, so there are 9 features that define cancers. Okay, so these are the features of cancers that allow them to behave abnormally, do whatever they need to do to survive and invade, metastasize. All right, so we'll start from the first one. Okay, so for number one, growth signal self-sufficiency. So what does that mean? Means the cell is able to self-stimulate itself or self-stimulate itself to keep growing in the absence of growth factors or growth stimuli. The cancer cell is able to stimulate itself, okay, to make its own growth signal to keep growing autonomously. Without the body telling it to grow, the cell is doing what it wants, it's doing something to grow by itself, to grow, to keep multiplying in numbers. All right. So, and what underlies that self or autonomous growth signal, that the cell is stimulating itself by itself to keep growing, right? We have mutations. So, like we said, this mutation, DNA mutations that underlie cancer, it can affect any of these nine, any or all of these nine signals, right? So when you have mutation in genes, we'll come to this, we'll describe these genes in more detail, but starting with proto-oncogenes, all right? So when we say proto-oncogenes, they are genes that stimulate growth, genes that stimulate cells to grow and multiply, all right? So when you have mutations in those genes, of course, the cell will enter an autocrine. When we say autocrine, that's a self loop of keep stimulating itself to grow, right? So some of the genes for that proto-oncogenes, they will produce, they enable the cell produce growth factors, where the growth factors will stimulate the cell. So if you have the malignant cell, so it will stimulate a growth factor, right? Release it, and the growth factor will keep stimulating that cell over and over and over again. So that's why they talk about the autocrine loop, right? So an example of a growth factor here is this platelet-derived growth factor in brain tumors. So some brain tumors produce growth factors that keep stimulating the same brain cell. So the brain cell produces its own growth factor to keep stimulating itself, to keep growing, right? What's another factor? So one thing is they produce growth factors. The other thing is what they do, they also produce growth receptors, growth factor receptors rather, right? So if you can imagine, we talked about the growth factor, producing the growth factor itself. Now you have some cancer cells who have the growth factor receptor that it can secrete to stimulate that cell to keep responding to the growth factors on the cell. All right, growth factor receptors. An example here is this HER2, HER2 in breast cancer. Okay, very important in breast. We'll come across it shortly after the today's break. Then other more signal more growth survival mechanisms are the signaling molecules such as RAS, transcription factors such as MYC, and cell cycle regulators, cyclin dependent kinases. Okay, so cyclin dependent kinases with respect to the cell cycle. Okay, when we talk of cell cycle, there are molecules that allow the cell to keep going through the cell cycle and divide continuously. Right. So the next step two, is anti-growth signal insensitivity. All right. So for the first table here, this first table, the most important gene I want you to remember about this is proto-oncogenes here. Okay. Proto-oncogenes. That's the most important thing I want you to remember from this first one. So when you talk about proto-oncogenes, when you talk about growth survival, increased growth in the cancer cell, what's driving it? Proto-oncogenes. Okay. And when they become mutated, they become oncogenes. When proto-oncogenes become mutated, they become oncogenes. All right. So that's the most important point I want you to remember for this first part. All right. What stimulates the cell to keep dividing? Mutation in proto-oncogenes, transforming them to oncogenes. That's the most important thing. For this one, for the second part, the next most important gene I want you to remember here involved with anti-growth signal insensitivity. What does that term mean? It means the cancer cell is not responding to growth inhibiting signals or signals that tell the cell don't grow, don't divide. The cancer cell is resisting those signals. And what gene is mutated to allow the cancer to resist anti-growth signals? Tumor suppressor gene. That's the most important point I want you to remember here. Tumor suppressor. So number one, the first one I said proto-oncogenes. Okay. Of all the things we're saying here, just remember proto-oncogenes. Of everything we're talking number two, just remember tumor suppressor. So if proto-oncogenes stimulate the cell to grow, tumor suppressor slow the cell down and tell it not to grow. So if the cell wants to keep growing, the cancer cell, what will it do? It will block or mutate or get rid of tumor suppressor genes. All right. So that's why they say mutation in tumor suppressor genes. Right. An important example of those are retinoblastoma. Okay, that's RB and P53. Those are the two most important or common tumor suppressor. We'll come across them in the future. So for step one, for the first signal, proto-oncogenes. For the second feature, anti-growth insensitivity to anti-growth signal, tumor suppressor gene. All right. So there's absence of tumor suppressor gene for in this one. There's increased activity of proto-oncogenes or oncogenes in the first one, right? Then the third one, evasion of apoptosis. Okay, evasion of apoptosis. So remember when we talked about apoptosis earlier, we said it's a form of cell death, suicide death, all right? And then when we compared it with necrosis, when we say necrosis, we're saying there are things that happen from that there are factors from outside the cell, external factors affecting the cell and causing it to die. When you talk of apoptosis, there are internal things that happen internally in the cell and causes the cell to die by to kill itself. So necrosis is external, apoptosis is something goes wrong inside the cell. Necrosis is something coming from outside the cell, affecting the cell and causing it to die. Apoptosis is something going wrong inside the cell, causing it to kill itself. All right, commit suicide. Now we talked about there are three main pathways in apoptosis. We said intrinsic, okay, involving the mitochondria, extrinsic, involving the death receptor, that's fast ligand fast, okay. We talked about the third one, that's perforin granzyme pathway. Of all those three pathways, the most important one is the intrinsic pathway. So when they talk about evasion of apoptosis, they're talking about a block in the intrinsic pathway. Okay, let's look at it shortly. The intrinsic pathway. All right, let's go back a bit. Okay, so we're talking about the intrinsic pathway here, which is this one. Okay, this pathway, that's what they're talking about here. All right, this pathway. So, and of this pathway, which one is the cancer cell most more likely interested in to help it keep surviving or if not die? BCL2, right? Remember we told you that in internal pathway, cytochrome C is a very dangerous molecule once it's outside the mitochondria. Inside the mitochondria is involved in oxidative phosphorylation, and outside the mitochondria, it will stimulate caspases that will activate enzymes that will digest the cell and lead it to die by apoptosis. So there are molecules that either allow cytochrome C to come out of the mitochondria or block it or keep cytochrome C inside the mitochondria. And those two molecules are these two, one, two. We have the BAX and BAC and the BCL2. Okay, so BCL2 prevents cytochrome C from coming out, while BAX and BAC allows cytochrome C to come out. So these two group of molecules are battling to keep cytochrome C inside the mitochondria. Now, in cancers, they found a way to delete this one and keep BCL2 constantly so active. So that means cytochrome C never comes out of the mitochondria. And so the cancer cell apoptosis will never be activated. All right. So there are even some tumors, like we've looked at before, there are some tumors like follicular lymphoma here, right? Follicular lymphomas where BCL2 is overexpressed. So BCL2, like I said, is anti-apoptotic or prevents apoptosis. So it will be helpful for the cancer cell to keep surviving by overexpressing BCL2 because BCL2 will prevent the intrinsic pathway. All right. So you can see here BCL2 overexpression, right, in these follicular lymphomas, right, which reduces caspase activation and allows the tumor regeneration. When you talk to tumor regeneration, that's just allowing the cancer to survive and multiply and keep growing. All right. So that's the same thing that I'm referring to when you come back to the hallmarks here. They say evasion of apoptosis. Talking of so there's mutation in the genes that regulate apoptosis. And what are the genes? They're talking of either getting rid of P53, because why is it getting rid of P53? Remember we said here in oops, sorry. Remember we said earlier that in P53, okay, in P53, what happens? P53 is going to support the activity of BAX. P53 is working with BAX and BAC to push cytochrome C out of this mitochondria so that it will activate caspases and kill the cell. So if the cell is either gets rid of BAX and BAC or gets rid of P53, then only PCL2 is allowed to work and so keep cytochrome C inside the mitochondria and allow the cell survive. All right. So when we talk of apoptosis, evasion of apoptosis, I want you to think of one thing here, BCL2. All right, BCL2, which is anti-apoptotic. And an example of a tumor that was expressed that follicular B-cell lymphoma. Remember we said lymphoma is a tumor of lymphocytes, right? So moving on to limitless replicative potential. So the most important term I want you to get from this fourth step here. Okay, so this is 1, 2, 3, 4. So the fourth step the most important thing I want you to get here is most important term I want you to keep in your head here is telomerase. Telomerase. So here we said proto-oncogenes. Okay, here we said tumor suppressor. Here we said BCL2. Okay, here telomerase. Most important term I want you to remember is telomerase. Okay, telomerase. And what are they? Telomerase, whenever a cell divides, right? You have your cell. Okay, your cell has chromosomes. The chromosomes are inside the nucleus. Okay, so as it's dividing, anytime it divides, right? There are telomerase are enzymes that act on telomeres. Okay, you say what are telomeres? They are enzymes that act on telomeres. Okay, telomerases are enzymes that act on telomeres. That's the simplest way I can define. And what are telomeres? Okay, so telomeres are they are DNA sequences of the ends of chromosomes. They are located at the ends of chromosomes. So if you have this is your centromere, okay, those ends of chromosomes are telomeres. So every time the cell divides, the telomeres act like a biological clock of the cell. So anytime the cell divides, what happens to the telomeres? They start to get shorter. No, they are located at the ends of the chromosome. So they start getting shorter and shorter and shorter. Okay. So they keep getting shorter and shorter. And then the next, up to a certain point, if they get to a certain length, the telomeres get to a very small length, just very tiny bits and pieces. Okay. What happens? When it gets to this point here, the cell will stop dividing. It can no longer divide. So that cell stops dividing and it enters what they call senescence. That is, it ages out and the cell will die normally. And that's part of the thing when we get to aging. These telomeres and telomerases and senescence and aging, they all come there. All right. So a cell has a limited time for dividing, cannot divide. A normal cell cannot divide forever and ever. Okay. They have those telomeres that shorten as the cell keeps dividing. So once it gets to the telomeres reach a certain length, the cell will stop dividing. Now, remember when we talked of labile cells, labile tissue like stem cells and all of that, they also have these telomeres inside them. And what does the telomeres do? It comes every time you divide, the cell divides, the stem cells divide. They'll come back and replace the part that was missing during each cell division. They keep adding those telomeres to the telomeres. So it allows the stem cells. So that's why your bone marrow stem cells can keep replicating all throughout your lifespan. because they have those telomeres that keep adding the ends of the telomeres at the ends of the chromosome. Same thing with cancers. They also have found a way to activate telomeres and keep adding, maintaining the length of telomeres, and therefore be preventing chromosome shortening and cell aging. All right. So number one, for the constant growth, like I said, proto-oncogenes, okay, increase the activity of proto-oncogenes. Number two, inhibition or blockage of tumor suppressor gene. Number three, evasion of apoptosis, increase the activity of BCL2. Number four, what? Activation of telomerase, telomerase enzyme. So number five, angiogenesis. When we remember we said angiogenesis in wound healing, angiogenesis just means new blood vessel formation. Angio vessel genesis, new. So new vessel formation, right? So from that word is self-explanatory. The tumor cell, why is it making you? The only question now is why do the tumor cells need to create new blood vessels? The singular for because if they don't, they cannot expand beyond a certain size. Right? If you don't, the tumors don't create blood vessels, they'll be limited in their size. Okay, they'll be limited in the size to which they can expand. So they need to make new blood vessels so that they can get bigger and bigger and bigger. So if you have this tumor here, if there are no blood vessels, it cannot expand to become this. So for it to get from this small size to this, it has to stimulate blood vessels to come and supply oxygen and nutrient. And also, not only in terms of increasing in size, it needs to stimulate blood vessels so that those blood vessels can bring what? Oxygen and nutrients to the, because the cancer cell is rapidly dividing. So they need nutrients, oxygen constantly. So that's why they stimulate b- blood vessels to come in and supply the nutrients and oxygen that they need to keep dividing and multiplying. So and what's the most important factor that stimulates angiogenesis for tumors? VEGF. That's vascular endothelial growth factor. Okay. So there's increased production of this VEGF. So when you hear the angiogenesis in tumors, I want you to think vascular endothelial growth factor. All right. And what is angiogenesis? New vessel formation. All right. Creating new blood vessels. All right. So for number six, Warburg effect. Now here we're going to jump into a bit of biochemistry. All right. A bit of metabolism, biochemistry. So when you think of Warburg, I want you to think of what? Glycolysis. Okay. Think of glycolysis here. So what's happening here? When you get when you talk of glycolysis, what are you talking about? Metabolism of glucose. Okay, you're talking about glucose breakdown into what? Pyruvate. Okay, and then eventually lactate. Now, when you talk about the breakdown of glucose to lactate, this is you're talking of anaerobic respiration. Okay, or anaerobic glycolysis. When we say anaerobic, meaning in absence of oxygen. But why cancer cells carry out this glucose breakdown to lactate, even in the presence of adequate oxygen. So what's happening in the cancer cells? You're having what? Aerobic glycolysis. So this is the central term. When you hear Warburg, I want you to think of aerobic glycolysis. So what's happening in aerobic glycolysis? The cancer cells are breaking glucose to lactate in the presence of oxygen, adequate oxygen. That's aerobic glycolysis. All right. Aerobic glycolysis. That's what they are doing. And why are they doing this? Because they are doing this so that they can get a lot of intermediates, or there's a lot of intermediates in the pathways to create DNA, RNA, proteins because they are rapidly dividing. DNA, RNA, proteins. They need a lot of intermediates, metabolic intermediates to create DNA, RNA, and proteins. Okay. This thing doesn't only happen in cancer cells, even in rapidly dividing labeled cells like bone marrow, and even during embryogenesis, it happens. But when you think of Warburg effect, I want to think of glycolysis. And what type of glycolysis? Aerobic, you know, aerobic glycolysis. Aerobic glycolysis. That's what I want you to think of. And why is it doing that? And what happens in aerobic glycolysis? Glucose is being broken down to lactate. And why is it doing that? Because it needs to create DNA, RNA, proteins for its because it's rapidly dividing. It needs to create DNA, RNA so that it can divide into two cells. Those two cells become 4, 8, 16, like that. Because that way that's how tumors grow, right? So that's for Warburg effect. Okay. Now moving on from there, so that's for Warburg effect. So that's what they are describing here. So you say a shift of glucose metabolism away from mitochondrial oxidative phosphorylation towards glycolysis, as we said, even in the presence of oxygen. And what's that term? Aerobic glycolysis, which provides cancer cells with carbon needs for synthesizing cellular structures. So the central term here is aerobic glycolysis. Okay. So now moving on to the next thing. Immune evasion in cancer. So remember we said what are the immune cells? The immune system we say has the innate and adaptive. Okay. And both these innate and adaptive are involved in detecting and destroying cancer cells. Right. So for the cancer cell to survive and grow and multiply, it has to find a way to evade and or dodge the immune system. All right. And what are those cells that are involved in catching and or attacking cancer cells? Number one, macrophages. Okay. Number two, CD8 T cells. Number three, natural killer cells. So these are three cells that go around looking for cancer cells to kill them, and the cancer cell has to find a way to dodge these three immune cells. So how does it dodge them? Three mechanisms. One, two, three. So for the first one, three mechanisms that it uses to dodge these immune cells. Okay. So the first one is what? Remember, when we say MHC1, which cell are we talking of? CD8. Okay. So if MHC1 expresses antigens to CD8, so the first mechanism that the cancer cell will use to dodge the tumor, to dodge the CD8 is it will reduce expression of MHC1. So if you reduce the expression of MHC1, that means cytotoxic T cell will not see it. All right, will not see it and will not recognize it and tell you to leave it alone. All right, so reduce the expression of MHC1, so it has taken care of CD8. Now remaining the other two, how will it deal with the other two? It will that comes to the second mechanism to produce immunosuppressive factors. All right, just like immunosuppressive factors, and even recruit T regulatory T cells or regulatory T cells. What do we say regulatory T cells do? They produce interleukin 10 and, more importantly, TF beta. These are immunosuppressants. So you see what it's doing? It now invites another person to come and block these three, to block these ones. All right. So that's by inviting Treg. So it will suppress the immune system. That's the second mechanism. So the first one, avoid CD8 T cell. How? By reducing MHC1 expression. The second one, invite Treg to suppress all the other ones, all the immune cells that will kill it. But Treg will come and block the three of them by producing interleukin 10, transforming growth factor beta. Then what's the third mechanism that to use to evade these three immune cells, right? It will upregulate checkpoint molecules that inhibit. We'll look at those checkpoint molecules in the coming future, but those checkpoint molecules. What do? They induce energy or immune tolerance in these immune cells. So the cells will see it, but they but they are not responding to it. All right. So those are three mechanisms. So one it will block CD8 T cell. Sorry, someone's mic is on. Please mute your mic.
Was not
Please
there?
mute your mic. Mute your mic. Someone's mic is on. Hello. Please mute your mic. All right. So number one, this will block CD8. It will block CD8. How does it block CD8? Reduce MHC1. Number two, it will invite Treg to come and secrete immunosuppressants and block the three of them. Number three, it will upregulate immune checkpoints. All right. It will upregulate immune checkpoint. What are those immune checkpoints? To give you a starter, that's the PD1, PDL1, CTLA4. Don't worry about them for now. Just know they are all immune checkpoints. All right. We'll get to them shortly. All right. So moving on from that. Okay. What's the next one? Number 8. Okay. Second to the last. So the number 8, tumor invasion. So talking about one of the defining features of a cancer or malignant tumor is invasion. The next one is what? Metastasis. So how does it invade? When they talk of ecadherin, what is it? Ecadherin is a molecule that holds the cell together. Just imagine two, like where you have a barricade of police officers holding a barrier against protesters. They lock arms together so that they form a wall against the protesters. So that's essentially like ecadherin just holds the cells together like in a line. So this is what ecadherin just holds them together. So that's ecadherin. Okay. So it holds the cells together in line and prevents them from moving away from that line of cells. All right. So if one of these now becomes malignant, for example, what will it do? It will first break that hold, to break the hold. Okay. Why is it not deleted? To break that hold, it will lose itself from the hold by getting rid of that ecadherin. It will delete it, get rid of the ecadherin, and losing that intercellular junction. So now that it has loosened it, then it's free to wander off. Free to wander off. All right. So this cancer cell is free to now leave that formation and go wherever it wants to go. That's the first sign that it's now, it was already, that all these other things that happen at the top have happened and transformed the cell, become a bad cell. Now it's ready to wander off, invade, and eventually find its way into a blood vessel and and travel far away to no man's land, to a distant organ. So, but the first thing you to do in order to begin that journey of 1,000 miles, imagine a cancer cell in the leg, in the skin cancer in the leg, and it wants to get to the brain. So that's the first thing. Number one, to first losing the junction between it and the neighboring cells. Okay, and once it losing that junction, what will it do? The next thing is it will now invade the underlying connective tissue. Okay, underlying connective tissue where it will produce, it will travel within that tissue. How does it move within the connective tissue to produce matrix or metalloproteinases? Okay, remember we talked in wound healing, we talked of metalloproteinases. What are they? They are enzymes that help to break down or degrade the connective tissue, or collagen in the connective tissue. So the cancer cells will produce that type 4 collagen. It's also another name for it is type 4 collagenase. Okay, so to produce it to degrade the basement and extracellular matrix so that it can move around. And while it's moving, how does it attach? It will attach to those extracellular membrane components and then move around, migrate, okay, through the degraded extracellular matrix. That's the form of locomotion. And then eventually to enter the blood vessel and then travel to distant sites. All right. So now, last one, metastasis. Metastasis is when this cancer cell gets into the bloodstream. So it has loosened itself. It has traveled through degrading the extracellular matrix and then penetrates a blood vessel. Once it enters blood vessel, it's on a highway to wherever the blood can take it. Right, travels as far as it can go. So they will travel via the lymphatics or the blood vessel and go to distant sites. Right. So sometimes you can predict where they can go based on the location of the where the primary site of the tumor. That's why I say site of metastasis can be predicted based on the primary site of the tumor. Right. So you can't predict it, but a lot of times you cannot. Right. And then they say cancers also show tropism for or certain organs. So some cancers prefer to go to certain organs, as we see, like prostate cancer, for example, goes to the spine. All right. Some cancers go to the brain, some go to the lungs. They have like lung cancers go to the adrenals, for example. There's no, they don't know exactly why they do that, but some prefer to go to certain organs than others. All right. So that's all for that.